All right, well, it's an honor to be here. Glad I made it on time up I-81. Yeah, so I'm at Virginia Tech Fish and Wildlife. My advisors are Dr. Angermeyer and Frimpong. And I'll move right in here. So a little bit of an outline. Let me get a little feedback first. Who knows what an IBI is in monitoring? A few hands, all right. How about who's done some electrofishing? A lot of hands. And how about environmental DNA? Who knows something about that? Okay, all right, thank you. Well, that's basically what I'll be talking about. For my PhD research, I'm looking at the effects of land use, particularly agriculture, on Chesapeake Bay streams and in relation to fish communities. So I wanted to check out environmental DNA as an alternative to electrofishing and conventional methods for sampling those fish communities in that context. And this is a work in progress, so these are preliminary things I'll be showing you. And they relate, I'm sorry, I got to stand in front of the monk. They relate to a case study on the Delmarva last summer where we collected environmental DNA from 29 streams and also electrofished the same streams. So when you see a stream like this, there's a little bit more than what meets the eye. So a lot of what's going on there in the stream, the depth of the stream, the height of the banks, the taste and smell of the water, a lot of that stuff is determined not by things that you can see on the screen, but maybe things that are upstream in the watershed, or maybe things that are out on the floodplain. And so you have this sort of looking at it a different way, a very complex way that fish communities or insect communities get assembled in a stream. There are processes that occur at very large scales over the region or the watershed, like the climate, precipitation, land use, geology, and those sort of things, that then exert influence on lower scale processes involving how sediment and water get into the stream, and shading of the stream banks, and how wood gets into the stream, what affects water quality, et cetera. And then all these multiple things are shaping the biota in the stream. And so sometimes a lot of streams, unfortunately, end up getting also influenced heavily by human impacts. And so we may need to be able to answer the question, how messed up is this stream in relation to other streams, in relation to natural streams that aren't so messed up? And with such a complex framework that we're working with, we often resort to sampling the fish community or the insect community to give us some insights as sort of a quick and dirty first look at what's going on in the stream. So then the next question is, if you're going to look at a community in the stream, what kind of animals should you look at? Do you want to look at fish or bugs, or do you want to look at algae? And all three are commonly used in all states. Macroinvertebrates are used for bioassessment, and fish in 2/3 of states, and algae in about 1/3 of the states. And that decision depends on sort of where you're at. In the Pacific Northwest, there's not a lot of fish diversity, for example, and so you're kind of stuck with other organisms. But in Virginia, as long as you're not in the cold mountain streams, you've got quite a few fish species to work with, and so that's an option. And then the second thing you might look at is what sort of questions are you asking? If you're asking questions about what is going on right here, right now at this site, maybe because there's a point source here or some kind of input right here, then you might want to look at insects or algae or both that are more sensitive to those proximal changes in water quality. If you want to get more of an idea of what's going on in the whole watershed or at larger scales across a region, you might consider using fish because of their larger body size, longer lives, greater mobility. They can reflect what's going on in a watershed pretty well. So this is just a look at a generic fish index of biotic integrity, or IBI. And I just wanted to point out that it's made up of individual metrics that each get a score. So like over here, score 1, score 3, score 5 for each of these metrics. Some of the metrics have to do with the occurrence of species. So like how many of a certain type of species occurs. Maybe some of those are sensitive species, others might be tolerant species. And then some of those metrics have to do with how many individual fish, maybe of a given species, are occurring. So you've got two different types of metrics that go into an IBI, at least two different types. And then you sum them all up and you get one number, the index, that says either this site is very poor or excellent or something in between. Here's an example from the Chicago area from the literature. They measured both invertebrates and fish, computed it in IBI, and they both showed the same thing with respect to urbanization. The dark circles are the Chicago urban area, and as you go to gray and white, the urbanization gets less and less. And you can see the IBI scores declining along that gradient. Okay, so from here on out, I'll be focusing on fish. So the question then is how to sample them. The conventional way in streams, wadable streams, is electrofishing, backpack electrofishing, which is what Josiah has on here, and Thomas. So we've got a crew of 4 or 5, or maybe more if we can get them, going out and walking up the stream side by side and shocking and netting fish. And then we might also do some seining in the pools, just as a second method, maybe catch some of those fish that got away from us. So some advantages of these conventional methods are that they are conventional, they are standard, they've been standardized and validated. There are some known relationships between how many fish you catch electrofishing and how many fish are actually in the stream, although that does vary by species and by environment. And you also have a clearly defined sampling unit. You know that you collected these fish from this 100-meter section of stream. You put a block net at each end. Fish didn't get in or out. So you're not, there's no ambiguity about where those fish came from. And of course, another advantage can be that you get fish in hand. And some of those metrics might require fish in hand, like if you're going to look at anomalies or diseases, or you want to get size structure or age structure, things like that on the population. And you don't see too many guys giving a smirk like that when they're collecting eDNA out of the water. It's a lot more fun to catch fish than eDNA. But there's a lot of limitations that this community has struggled with over the years. It's very laborious, so it takes us about 6 hours to do one site. And that's with 4, 5, or 6 people. So to do 29 sites takes a whole month. And then your sample unit is small. Because it's so laborious, you can only sample a fairly small section of stream. That's usually based on the number of channel widths, so maybe 20 to 40 channel widths might be one section of stream, which we call a reach. And you might go 2 passes through that. And then you might go do a second reach and do 2 passes through that, and that's why it takes 6 hours. It can be hazardous. You're working with electricity and water. And it's invasive in the stream. The equipment's expensive. And you still don't get perfect detection of even what's in that reach after all that work. You might detect 90% of the species. You got to have a good taxonomist that knows how to identify the fish. And you just can't get to some places. Really, anything deeper than your waist is pretty hard to sample. And places that are choked full of wood or quicksand or things like that. So I wanted to look at it, environmental DNA metabarcoding as another way to sample fish. It's not a new way anymore. I probably shouldn't have said that, but it's come on since about 2012 or so in the fish community. So you start by filtering some water, and this is not an electrofisher. This is an eDNA sampler. It's basically just a pump in a backpack, and it has a filter on the end here. And so this allows you to walk through the stream while you're filtering the water. And that part of it takes, from start to finish, well, we're filtering water for about 3 minutes and getting from 1 to 3 liters of water. Okay, and then you take that filter and send it to a lab and extract the DNA. You gotta choose a barcode from a gene that you want to use, and it's got to be one that works good for your community of interest. So there has been some work in developing barcodes for fish and other organisms. So you amplify that section, that barcode, you sequence it, and then you get something that looks like that as a deliverable. So you've got each row is a different sequence that was detected, and then you've got these columns over here are sites, and the numbers are called read counts. And the read count is here. It's just the number of times a given sequence was detected in the sample. So you could have multiple sequences that correspond to the same species, and that's what we found. So some advantages: it's really easy, very little effort. It's very cost effective. I could go out in a day, 2 days, I could go out and sample all of those streams that it took us a month to do with the electrofisher, and it would just be me, not 5 people. It's very easy to standardize at least the stuff that's going on in the field. You get a filter with the same pore size, and you set things up the same way. It's safe, non-invasive, very sensitive. You can catch a lot of species this way, or not catch, but you can detect a lot of species. You can identify them accurately. You can sample any habitat the water flows through. If you can't get into the pool, you can sample the water coming out of the pool. And it could be an advantage or a disadvantage. Your sample unit is probably larger in flowing water, but the disadvantage is you don't know how large it is. You have some idea. But in a small stream that's flowing slowly, that DNA might go maybe 1 kilometer downstream. In a large river that's flowing swiftly and maybe cooler water, it might go 100 kilometers downstream, and you can still detect it. So that's an area of a lot of research. So yeah, that's one of the limitations. The DNA is carried downstream, so you have sort of a fuzzy sample unit. You don't necessarily know the upstream extent or how long. Well, so you have an idea of the temporal extent there. Usually, I mean, that varies also, but you're talking about DNA in the water column that's degrading, and it's probably not going to last more than 60 days or so, and it may be a lot more recent than that. And then the amount of DNA will depend on the species and their behavior and the environment, so it's a little bit difficult to standardize in that sense. Okay, then there's some methodology that's in flux, depending who you ask. Somebody will say use a 1 micron pore filter, and somebody else will say use a 5 micron pore filter, and have data to back that up. When, how, when should you sample, where should you pull the water from, et cetera. Those are all open questions. What primers, what barcodes, how to do the sequencing and processing, where do what do you say is a detection and what's not a detection, those sorts of questions. And then a big deal is, in order to identify these unknown sequences, you have to have a library of a barcode library to compare them to. So if you're just relying on publicly available data, you may have a hard time identifying some of those sequences. It's best to have a local library that's been compiled from your watershed, your region. And then even though you do the best you can, you get the best barcode you can, you're still not going to be able to separate some species because they might have the same sequence for that barcode. Okay, so so far there's not a perfect primer set. And so sort of the result of some of those things is you can get good detection, but your abundance data has a lot of noise, sort of to say the least. You don't really know what the read count means. And I wanted to explore that a little bit. This is just a look at the number of articles that have been published since 2010 that have looked at estimating fish abundance using eDNA. And so you can see that it is very an area of interest. Now, 90% of these found a positive correlation between eDNA read counts from metabarcoding and abundance or biomass from conventional surveys. So there is some indication that you can get some idea of abundance from this. So last summer we went to the Delmarva and we sampled with electrofishers and an eDNA sampler, 29 streams. So just for this talk, I have some very simple hypotheses I wanted to look at. One is that eDNA will detect more fish species than the conventional methods, which I'm calling CES. And two, that the eDNA read counts will be positively correlated with the electrofishing fish counts. So I kind of already described how we did that. With the electrofisher, we did 2 reaches that were each 20 channel widths long, so that's an average of about 80 meters, and we sampled them each with 2 passes. And then we seined the pools after that. And with the eDNA machine, the eDNA sampler, we filtered 2 reaches per site, 1 upstream and 1 downstream of those electrofished reaches. And we tried to get at least 1 to 3 liters of water that we filtered through a 5-micron pore filter. And then we did the metabarcoding with this MyFish universal primer. We sent the filters to a commercial eDNA lab, Jonah Ventures, and they took care of the barcoding and sent us the deliverable with the read counts. And then we also were fortunate that worked with the Smithsonian Institute and were able to cross-reference against their barcode library for the Chesapeake Bay watershed. So a lot of pretty much all the species that we were finding were in that library. So this is just looking at the advantages of using that regional library as opposed to the publicly available barcode library on GenBank. So that deliverable report included 503 distinct sequences from fish, and a few from humans, and a few from cows, and a few from bats, but mostly fish. So of those 503 sequences, when we're using just the publicly available GenBank library, we're able to identify 360 of them to species, so only about 70%, and 440 or 87% to genus. But when we matched those sequences up with the Smithsonian's Chesapeake Bay library, we're able to get much better, much more of those sequences matched to a known fish. 91% were matched to species, and 100% matched to genus. We noticed that there were a couple strange IDs in the GenBank results. Not too bad, but mud sunfish that were confidently identified as rock bass, which don't occur there, and red ear sunfish reported as red spotted sunfish. Okay, so looking at which method detected more species, the first row is showing that the conventional methods detected 40 species total over all the sites. And the eDNA detected 36 species plus 2 genera that couldn't be resolved to species. And those 2 genera each represented 2 species that were present on the Delmarva. So we sort of roundabout got 40 species, but only able to identify 36 to species. And then each of those methods detected 3 species that the other method did not detect. And then if you look at the average number of species that were detected per site, we saw that eDNA detected almost 17 species per site and electrofishing about 15 species. And that was a significant difference. And then the flip side of that is how many sites had more species detected by each method. So 18 of the 29 sites had more species detected by eDNA, and 5 of the electrofished sites, or 5 of the electrofished samples had more species detected by electrofishing. And I just wanted to point out that that barcode that we used, these are the 2 the genera I mentioned before that were not able to be resolved. The pickerels and the banded sunfishes. So this just looks at if we had only sampled with one approach, how many or what species would we have missed? So you can start to get a feel for which species is it working well for with eDNA versus electrofishing. The one standout is the least brook lamprey here. The number across the top represents the number of sites where they were detected out of the 29 sites. So those lampreys and the stacked bars here represent what proportion of sites eDNA versus electrofishing detected a species at. So the least brook lamprey were found at 19 sites, and I think it was all but one of those, they were found only by electrofishing. So their DNA wasn't up in the water column, and there's probably a good reason for that. But over the rest of the species, eDNA outperformed electrofishing. So all the bars that have sort of the orange yellow that's longer than the purple is where eDNA did better. And this is sort of looking at the flip of that. The x-axis is the sites, and the y-axis is the proportion of species at a site that were detected by either method. The green bars, which I should have mentioned before, the green bars are where a species, in this case a species was detected by both methods. So there's a lot of overlap. They're both sampling the local fish community. But again, eDNA has the longer bars on 18 of these sites, and electrofishing has the longer bars on 5 of the sites, as I mentioned before. This is just a quick look at that second hypothesis, that there would be a correlation between, so this previous stuff I was looking at, this is all has to do with just presence or absence of species. Now this one is looking at abundance, or those reed counts, and the number of fish that we caught in our hands. So there was a positive correlation that was significant there. There's also a lot of noise. Down here, the electrofishing counts, there were some sites there where we caught some fish with the electrofisher where we didn't find that species with the DNA, and that's probably mostly those lampreys there. And then there were a lot more cases where we detected some species with eDNA that we did not catch with the electrofisher. So going forward, I'd like to just dig into the weeds a little bit further with these numbers, see how it breaks out by species and by site, and see if there are environmental variables or species traits that explain some of this variation. We're also going to try and, well, actually already did it this past summer in another set of streams on the Piedmont, so I can get a replicate. Those filters are still sitting in a fridge until the USGS sends us some more money. Let's see. I want to check biomass, not just eDNA count, or see how biomass relates to the eDNA counts, not just the number of fish that we caught with the electrofish here. And again, I already mentioned looking with the effects of some environmental factors and species traits on that correlation between eDNA and electrofishing. We also collected those upstream and downstream eDNA samples. I'd like to use those to get a feel for are these counts changing as we go from upstream to downstream in relation to what the electrofisher says is in a site. And maybe I'll compute some IBI scores from both methods and see how they compare. So I would like to acknowledge Greg Noe at the USGS, who is the leader on this larger project and did all the site selection and got permission with his team for all that. And Rob Aguilar at the Smithsonian was the guy that made that barcode library available to us. Joe Crane at Jonah Ventures, we had some discussions with him about the results. And Aaron Malloy with the Fish and Wildlife Service has also been giving me a lot of tips on metabarcoding, so very much appreciated. And that's not mine.
Any questions?
Thanks. So you mentioned the barcoding libraries, and I just wondered how prevalent those were across the United States.
Sorry, could you repeat that?
How prevalent are these barcoding libraries across the United States?
How many barcoding libraries?
Well, I mean, are they just readily available for you to source the information from? I mean,
yeah. So the Smithsonian has a website, linking to this, but it's not publicly available yet. So I had to work with them and send them the information. I don't know a lot about other, regional library efforts that are going on around the U.S. I imagine there probably are several, but I've only been working in the Chesapeake Bay watershed on this.
Do you have a theory as to why the brook lamprey wasn't detected in the eDNA? Was it not
A theory?
The lamprey, why that one wasn't in the eDNA?
Yeah, so we were there in June, and those least brook lamprey pretty much live in the sand, down in the sand. When you electrofish, if you wait a few seconds, they might come boiling up out of the sand, but it takes them a little bit, so they're down in there pretty good. And I think, they spawn in the spring, and then they don't live long after they spawn. So I think it's mainly because their DNA is just in the sand, not in the water so much. But we do have some samples we collected in April that we were going to look at macroinvertebrate eDNA, that I could potentially go back and pull some of those and look for fish eDNA and see if the lampreys are showing up then when they're spawning.
I was just curious if it would be safe to say if the eDNA does a better job of getting the the watershed characteristics of the fish community, since the eDNA takes time to travel down, whereas the shocking is just more of like a localized community.
Yeah, that's kind of exactly why I wanted to use eDNA was to see, and I don't have a very good design for answering that question. But But what I'm thinking is exactly what you said, that yes, the eDNA is sampling a larger sample unit, and therefore it should better represent or better respond to the conditions throughout the watershed. These are all small streams, like 10 to 50 square kilometers, so they don't have a very large watershed. So the DNA should be sampling is definitely sampling a larger portion of that watershed than just, our 2 80-meter reaches. One way I want to look at that is by, compiling a big matrix of all the environmental variables I can get my hands on and seeing how much of the variation in the eDNA read counts that explains versus the electrofishing counts, and basically see which are better correlated with the watershed conditions.
Here we go.
But I'm also open to ideas on that.
I was just curious how turbidity affects the eDNA readings.
Oh, which?
Turbidity.
Turbidity. I don't know a lot about that. I did see one study by Jamie Roberts, who used to be at Virginia Tech, looking at a single species Roanoke log perch in the Roanoke River, and he did not find any effective turbidity on detection of Roanoke log perch using eDNA. He didn't find any effective turbidity or temperature or pH. I don't remember what all he measured, but.
So it doesn't affect the extraction process or the filter?
So it definitely affects the filtration, and so you might have to use more than one filter. We found that we're using a fairly large pore filter of 5 microns, whereas others might recommend 1 micron, and we're still clogging that up pretty quick in a muddy stream. And so we might run another filter through if we need to. And it makes a difference whether you stir up the mud a little bit. You want to get that while the water's clean. So, yeah.
Any other questions?
Hi there. I was just wondering, is there a varying half-life to different DNA from different species of fish?
Varying what?
Like a half-life on how DNA breaks down between different species?
Yeah. So I mentioned some rule of thumb I came across in the literature was that in a stream or river, you might expect the DNA to last like 60 days or so, or up to 60 days. But in sediment, it lasts for thousands of years. So if somebody's stirring up sediment, you might be picking up old DNA. But in terms of by species, I don't recall seeing anything on that, at least not for species that I'm working with. But that's one of the things I want to look at is how do those eDNA counts relate to the electrofishing counts by species? What are the traits of species where that relationship is very close, and what are the traits of that species where the relationship breaks down, like the lamprey? Yeah.
Any other questions? Last call. Oh, yeah, try it.
Does it work this time? A little bit better. So with you talking about bringing up more sediment into the water column, potentially increasing DNA, how accurate do you think it is? I guess how do I rephrase this? Do you think the number of species you're getting being higher for that sampling versus conventional methods, electroshocking, do you think that's accurate, or is it old stuff that's no longer there?
Right. So I can't really answer that with confidence, other than to say studies have shown, in the water column it doesn't last very long, but in the sediment it does last very long. So, if we went up behind a herd of cows that was stirring up the sediment in the stream, that probably wouldn't be a very reliable sample, and we wouldn't be able to get very much water filtered. But typically, we're sampling at low flow, the water's clear, and we're not disturbing the substrate, and so I don't think it's much of an issue under those conditions. Yeah. Yeah.
All right, last call. Okay.
Thank you.